This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 484, recorded on March 9th, 2018. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me here in New York City, Dickson Despommier.
Hello, Vincent.
Where it is, what? Nice, not so nice.
It's pretty brisk out there right now.
4 degrees Celsius, cloudy. We just got through a big snowstorm.
We did.
Where I lost power
for
24 hours. How about that for a poem? I lost power for 24 hours. Now, Dickson, you said the episode 484.
It's an interesting number, that's all. You know, you can divide by 2, divide by 4, and you get a palindromic numerical, 484, that's all.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey.
Here it's 30 degrees Fahrenheit, which is -1 Celsius. And it's cloudy, and it says we're going to have snow starting in 46 minutes.
Oh, my goodness.
I'll have to watch.
There you go.
Now, what have you got on the ground now?
Oh, we don't have really anything.
A dust, not even a
leftover
dust.
stuff.
Okay.
I was listening to a podcast this morning on the way in, and they're talking about the temperature, and one guy said, "Don't tell me that stupid Celsius stuff." Can you imagine? He called it stupid Celsius.
Stupid Celsius.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. I remember a time during TWiV when we kind of made fun of Celsius.
Yeah, but I only use Celsius, so...
Yeah, we came around on it. So, yeah, it's 36 Fahrenheit, 2 Celsius here. It's not bad. It's a little overcast, but the snow, we got about 8 inches of it, and really wasn't a big deal. The streets are practically dry at this point. So,
yeah.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, everybody. It's great to be back.
You got a lot of snow out there.
We did get a lot of snow. So we got about 23 inches of snow in the most recent storm. Although it's 40 degrees Fahrenheit now and
melting.
sunny out.
So Brianne is about 30 minutes from my home domicile.
And how much snow did you
So we got maybe 11 inches.
That's incredible.
Isn't
That's
that incredible?
Twice as much.
Yeah, it's a narrow band of something or other.
It just depends on which band of the
that's right, the northeaster.
The storm you're in.
So what is it? Wednesday morning, right?
Yes.
I wake up, there's nothing happening. So I come in, Columbia's on, right? Everything's open.
Looks easy.
Then it starts snowing like crazy. At 1 p.m. they cancel. I came to teach class, and they canceled classes at 1:30ish, 2 o'clock. So, oh well.
Yeah, we were also closed on Wednesday, and I sat at home and saw no snow in the morning and thought it was a big bust.
Yep.
Yeah,
not
right.
so much.
Right.
Wasn't a bust. All right, we have a few things for you. We have many things for you.
We have a plethora of things.
First, I want to tell you about the upcoming meeting called ASM Microbe, the annual meeting of the American Society for Microbiology, at which I saw Brianne, was it last summer?
Yes,
in
I
New
was
Orleans
there last summer.
with your lab.
Yes.
2 people from your lab, I think, or 3.
I had 3 from my lab last year.
And you all got black lab coats, I remember.
We did. My whole lab has matching black lab coats.
That's cool.
And everyone else here is very jealous of us.
They're really good. Anyway, this year it's in Atlanta, Georgia. And ASM has a special opportunity for our podcast listeners. This is not food.
No.
This is not hard drives. It's not videos. What it is,
It's not like Costco.
Get $50 off registration for Microbe 2018, which is June 7th through 11th in Atlanta, using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial scientists in the world. Delve into your scientific niche in 8 different tracks. Don't miss this opportunity. Visit asm.org/microbe. That's asm.org/microbe and use the promo code ASMPOD. It's all one word for $50 off registration. When you go to register, you will see, do you have a promo code? You type in ASMPod. Then you get $50 off, and ASM realizes that we have 1 billion listeners.
Yes.
And they're going to lose $50 billion
They've already budgeted for that, I'm
on
sure.
registration. Well, not 1 billion people go, unfortunately.
Yeah.
I don't know how many people go to this thing. But
It is a big conference.
Yeah, it's very big. You can not see someone there.
Yeah.
Right.
They can be there, but you cannot see them.
Yeah.
Really? You were there the whole time? I didn't see you once. See you in Atlanta.
I have an announcement about the ASV meeting, American Society for Virology meeting at the University of Maryland, the 14th to the 18th of July. New incremental piece of news, the on-campus housing registration is now open. So they have a lot of varieties of rooms you can get, singles, doubles, suites, etc. And so they expect those to go early. So for the best selection, you should register now. And the hotel rooms are also, there's a set of hotel rooms in 4 different hotels that are blocked off. So there's a map at the bottom of the lodgings page on the University of Maryland ASV 2018 website. And so you can see where the dorms are, where the hotels are, and so forth. But want to do that pretty soon.
And that website is asv2018.umd.edu, right?
Yes.
Okay.
And this is the main campus of
College Park.
College Park.
What do you mean main campus?
Well, there are several campuses.
Where are the others?
Yeah.
Where are the others?
The ones in Baltimore.
Baltimore, right?
Yep.
Yeah, I went to Towson State University at the time, which was the University of Maryland campus in Towson.
There you go.
There's University of Maryland, Baltimore County. There's University of Maryland, Salisbury. They're scattered around the state. But the main U of M campus is quite lovely. Classic college campus look and big.
Mm-hmm.
Isn't that in Silver Springs? Isn't that the name of the town that's
College Park?
What is Silver Springs? Where is that next?
Silver Spring
is out.
Only one spring, not multiple.
Silver
Right.
Spring.
So Silver Spring is also a suburb of DC, but slightly different direction.
Oh, okay.
You can get there on the red line.
All right.
Yes.
Alan, can you take that first follow-up?
I can. I got this email from Tom in Austin. It says, Dear Alan, as you enumerated the challenges to implementing the above, and he's referring to the science standards that was my pick last week, school science standards. I have a somewhat different take, at least as far as Texas goes. In this state, we have over 1,000 school districts. My employer advocates for these districts' collective agendas every legislative session. The fact is that you can't easily herd these 1,000 plus cats, but that situation helps protect us from some, at least, of the Texas legislature's attempts to make sure that no Texas school graduate knows how to think critically, especially when walking into a voting booth. And that is an excellent point, Tom. This is a two-edged sword that we can't have a national science curriculum because of this balkanized system, but on the other hand, it's also it limits the damage that could be done by misinformed folks in high places.
Yeah.
We have the same problem in New Jersey. We have many, many school districts, 678, and each has a superintendent, which makes over $150,000 a year. Isn't that crazy?
Really?
Yeah.
Wow.
I lived there all my life. I never knew this.
It's crazy. Totally crazy.
That's Massachusetts. It's similar.
And New Jersey is slightly smaller than Texas, as I recall.
Yeah. Well, they have over 1,000 school districts.
That's incredible.
We're going to go there this summer. We'll take care of it.
Wow.
Right.
Oh, well, yes, that's right. We will take care of it.
All right. We have Bob writes, at strategic points in TWiV 483, Rich Condit paused the discussion with a nod, quote, to the uninitiated, end quote, then proceeded to describe concisely various terms and techniques using non-technical language immediately understandable to this listener. One would hope this gets to be a permanent feature on future TWiVs. The panel doesn't deliberately try to be opaque, but sometimes it just happens simply because this is experts talking to experts. Keep up the good work, Bob. I like that little phrase, experts talking to experts.
Right.
Well, with the exception of me.
And, well, me.
Well, it's probably me.
I'm just kidding. We are. That was the whole idea originally behind TWiV. Virologists talking amongst themselves. I don't know where you came in, Dickson, but it works.
At the bottom of whatever you're talking about.
Kathy.
Sorry.
She's muted. She was muted.
Theo.
Theo writes, Hi, TWiVers. I'm listening to TWiV 483. Vincent mentioned that the eLife article he downloaded was branded manuscript submitted to eLife, and he seemed to suggest that the article might have been online before it was accepted. I just want to clarify this is not the case. The article had been peer-reviewed and accepted by eLife before it was made available on their website. However, it had not been edited into the final publication format, and hence was simply the document originally submitted by the authors. In this case, the author opted to use the nice latex template provided by eLife. He gives a link to that. More on the publishing process, and he gives a link to that. You can still access the original version of the article even though it has been edited since. The links are available in the section Article and Author Information under the heading Publication History. And he gives links to the archive page and the current page.
Cool.
Okay, thank you, Theo. I still think they should make sure that it doesn't say submitted when you download it, right? Because it makes people think without going. So we were talking about it, and I couldn't possibly have found this stuff right at the time. So, and others too. But thank you. That's very good. I didn't know there was a template. Cool. I can't afford to publish at eLife, so I never looked. Why do we have to pay to publish paper? Never mind. That's a different story. Let's talk about a paper in a journal where you have to pay to publish.
You do.
What's that?
Yes, you do.
Yeah, in Nature Communications. Yep. It's all the Nature journals you have to pay.
Now, you have to pay to publish there, but the upside of that is that the resulting paper is open access. So the rest
this one is open access?
To read it. Yep.
It is.
Oh, it says open, yeah.
Nature Communications is their
I didn't know
access
that.
effort. Yep.
But last night I'm writing a blog post. I needed a picture of these fuzzy viruses, and I go, crap, this is Nature. So I looked elsewhere, but I could have used this one.
You could have just clicked the link.
But I found actually a blog post by one of the authors where he had put on one of the EMs raw out of the EM machine, with all the magnification on it and everything. And that works because he dyed it blue. It was kind of pretty.
Oh, cool.
Because we know,
I like that image on your blog post.
Isn't it cool? Yeah, it was from Jonatas Abrahao, but I don't say it right because I don't know Portuguese. I'm sure the little squiggle over the A, or something like that.
Yeah. Mm-hmm.
Anyway, this is tailed giant Tupanvirus possesses the most complete translational apparatus of the known virosphere. And the first three authors contributed equally: Jonatas Abrahao, Lorena Silva, and Ludmila Santos Silva. And then we have a couple of people from France, Didier Raoul and Bernard Lascola. So this comes from Aix-Marseille University, Universidad Federal de Minas Gerais, Universidad de Brasilia, the Laboratory of Biomass Conversion. Cell biology.
That's a great name.
Biomass conversion. Cell biology and metabolomics platforms. INSERM in Paris, Université Paris Descartes, Université Pierre and Marie Curie, Hôpital Européen Georges Pompidou, and the Karolinska University Hospital. What a nice...
At CNRS.
Yeah. Too many.
Yeah, a lot of collaborators.
Such a nice diversity of locations, right?
Yes.
You bet.
And how would you like to go to the Pierre and Marie Curie University? That would be hot, wouldn't it?
Yeah.
No pun
Sounds good to me.
No pun intended.
Giant viruses. We love giant viruses. Well, at least I do. Does everyone love them?
Yeah.
I think they're very cool.
Okay.
And these are particularly neat because of the way they look.
So we have talked about giant viruses a lot on TWiV. In fact, not too long ago, Bodo Saltan's virus, right? Mm-hmm. And that one still hasn't been published, but we talked about that in a bioarchive preprint.
True.
And so this is another giant virus, but it's still, whenever a new one comes out, it seems we learn a little bit more. And a couple of amazing things about this one. So where did these come from? They have two Tupanviruses from two different places, soda lakes in Brazil. And these soda lakes have high salinity and pH, very extreme. You wouldn't want to go there. You wouldn't want to swim in it, I guess, right?
They might be neat places to visit. I put in a link for the Wikipedia article on soda lakes, and I didn't know there were any in South America, but apparently there are, and they're in Africa, and I think there are a couple of them in California.
They have a lot of them in Bolivia.
Oh, Bolivia. Okay.
Dickson,
tons of
have you been?
Mm-hmm. Yep.
Can you fish in them?
No, but the flamingos are there, and they're fascinating to watch.
They feed in the lakes?
They do.
Wow.
Yeah. They feed algae.
They eat spirulina algae.
There you go.
And they become pink, right?
They do.
Like makes them pink.
That's right.
So that was one place, and the other is a deep ocean sediment off the coast of Brazil.
Very different environments.
Quite. And
although chemically, I guess they might be kind of similar. I think the soda lakes are saline.
They are.
3,000 meters.
3,000 meters is deep.
A lot of pressure, right?
Very deep.
Yeah.
Mm-hmm.
So they must have sent something down to collect it.
Yeah.
So they found a couple of two different viruses, but they put them in the same group that are able to infect Acanthamoeba castellani, who was a mafioso from Hoboken, and Vermamoeba.
Don't mess with him.
Vermamoeba
He'll poke your eyes out.
vermiformis. Yeah.
I was going to say that Tupanviruses were named after a South American Guarani indigenous Indian tribe's god, Tupa, who was the god of thunder, one of their main mythological figures.
Yeah, it's a nice name, Tupanvirus.
Yes.
And
So if you get infected with one of these viruses, they leave you a little Thor.
yeah,
Mm.
that's right.
It's too bad they don't infect long-billed birds. They could have been called toucan viruses.
Yeah, toucan, right. Anyway, the capsid looks like a mimivirus capsid. It's fuzzy. But then these have tails, cylindrical tails attached to the capsid. And
They look like little bottles or light bulbs.
they're beautiful. They're just
lovely.
beautiful.
Really cool.
They're even
Yeah,
Okay, I'll go to the paper.
the tails are fatter relative to the capsid than any of the tailed phages that you think
Right.
of.
Yep.
They stand on stalks, kind of, but these are standing on these cylinders.
Yeah, they really they look like round bottom flasks in a
Yeah.
canister.
very pretty.
They do.
Florence flasks.
That's
Yeah,
what
you
they-
remember
okay.
Florence flasks
Yeah,
from
that's
Erlenmeyer
right.
flasks.
Yeah, they do.
The Florence flasks have a round bottom.
They call them the tail, the longest tail described in the virosphere.
They're comet viruses.
And it's not tightly attached, right? And this is interesting because there's a lipid membrane inside the capsid of these viruses.
Is that a thing? I mean, the membrane inside the capsid?
Yeah, it happens in other viruses, yeah,
Okay.
for sure. And then they say that there's something in the tail, right? You can see in the EMs that there's something in there. I don't know what it is. They say the tail content appears to be released after an invagination of the phagosome membrane inside the tail. So these are taken up into cells by phagocytosis, and they end up in a vesicle in the cell. And they have a picture here of the membrane of the phagosome poking into the tail, and it's somehow releasing what's in there. But then there's another picture where they show the head fusing with the phagosome, and presumably the DNA is in the head. So
yeah, those pictures are really cool.
I don't know what the tail is doing. I have no idea, right? Because it doesn't look like the DNA is going out through it. So it's doing something else, which remains to be
fascinating.
Maybe delivering some protein payload to the cell to prep it for the virus.
Maybe a transcription protein, right?
Maybe.
Because it's going to be setting up shop in the cytoplasm. Could be. Well, they say later they did proteomics on the particles, and they got 127 proteins. And half of them, they had no idea what they were. They didn't resemble anything in the databases. Yeah.
I like that they referred to that as orphans.
Yeah. Orphans.
Right.
I had never heard that before, but that I thought was pretty funny.
Orphan proteins.
The ORF, open reading frame, ANS.
Yeah. It's a cool name. And this virus genome, 1.5 million base pairs of double-stranded DNA, the fourth largest viral genomes described to date.
Yep.
I think the Bodo-Saltans was fourth or fifth. Bodo-Saltans is 1.5, I believe.
Yeah, and just to put this all in perspective for people who don't listen to the show all the time, I mean, normally you look at a proteomic profile of a virus, you get like 4 proteins or 6 proteins or some handful of proteins because it's not relying on proteins to start its life cycle. And they're much, much, much smaller in the genomes too, like thousands of bases instead of millions.
So presumably some of these proteins in the particles are in the tail there, and maybe they're, like you said, Alan, they're bootstrapping the infection. So we have 1,276 and 1,425 predicted open reading frames. And as Brianne said, some of them are orphans. 375 and 378 are orphans.
Mm-hmm.
I see a new name here, the rhizome of Tupanvirus, which is a graphical representation of gene-by-gene best hits. I've not heard rhizome before, and it made me think of...
It's a plant thing.
Yeah, plants.
Yeah. I'm not sure I'm a fan of this term. Because it's borrowing something from a completely different branch of biology and putting it to a completely different use. And I think that is setting things up for confusion.
Here.
Anyway, this looks like it's a mimiviride. All right, so it's in that family. And however, 775 of the predicted genes are not present in other mimiviruses. So this is quite diverged.
And they put together a phylogenetic, what is this, a phylogenetic shrub or when it's circular?
A phylogenetic shrub.
Do they have any metabolic genes?
They do not have genes involved in glycolysis, the Krebs cycle, and the respiratory chain.
Good, then it's still a virus.
Right. So I think that's an important point because they make the point that Vincent also does in his blog post that only the ribosome is lacking. Energy generation is lacking as well.
Sure.
Right. But I think that the point there is also that there are prokaryotic cells that are missing energy generation genes.
Yeah, they're considered alive, right?
They're considered alive, and so that difference is really ribosome.
Are those ones you're referring to just now, are they culturable?
So this is things like rickettsia and chlamydia.
Oh, okay.
So the obligate intracellular parasites.
Okay.
Yeah, this. Go ahead.
I was going to say, back on the term rhizome, I have the feeling it's because of the way the graph
looks,
chart ends up looking, and that's resembling a rhizome of a plant.
Yeah, maybe that would be fine.
But what's going to happen when somebody finds a giant virus in an amoeba that infects plants and
exactly
plots the rhizome? And what are we referring to?
That's right.
I don't know.
It says, in botany and dendrology, a rhizome is a modified subterranean stem of a plant.
Right.
Yeah.
This paper made the news the past couple of weeks, and a lot of the headlines were it redefines our notion of what is living, and I don't think it does at all.
No.
I spoke to a Newsweek reporter about it, and her headline, which she didn't make, of course, was it redefines what we think of as living. And I told her, I said, it does not. But she didn't listen. So the interesting thing here, it has lots of open reading frames encoding proteins that look like they're involved in translation. 20 amino acyl tRNA synthetases. That's it. That's all you need. 20. I mean,
and those are the enzymes that put the amino acids onto the tRNA, and the tRNA with the amino acid attached then is what connects with the messenger RNA to make proteins.
Right.
They have a lot of tRNAs, 67 and 70 tRNAs with 46 and 47 codons. They have initiation proteins. They have elongation proteins. They have termination proteins. They have lots of genes involved in tRNA maturation. It's just amazing. They say, as Kathy said, only among all the translation proteins, only the ribosome is missing. But of course, only is a funny thing to say about a ribosome, right?
Right.
It's pretty big.
But this is, I mean, the point that they're making is that this is really kind of insane for a virus to pack this nearly complete translational machinery on its way into a cell that's got all this stuff presumably already. Whereas most viruses would just exploit what's there, the mimivirus seems to have thrown out the whole script and said, okay, we're just going to bring everything.
So that's an interesting question. What is the function of having all of these?
What's the advantage?
So here's the experiment that you should do then. You should infect the acanthamoeba with the virus and then enucleate the acanthamoeba and see what happens.
Well, the virus essentially does that. It destroys the nucleus during infection.
But it must utilize the material from the nucleus as its own substrates.
They say that the ribosomes are cleared from the cytoplasm.
cleared.
They're wrapped up in little membranous packets. That's pretty weird. Now, this virus does not encode ribosomal RNA. They found some intronic ribosomal RNAs. In other words, in the ribosomal RNA genes, there are introns, right? And they found those, but no exons, none of the sequences that would be present in a ribosome. So I don't get why you would clear all the ribosomes from the cytoplasm. I don't get why you would encode all these proteins.
Sure.
I could see encoding a few tRNAs that maybe because your codon usage is slightly different from that of the host cell.
Right.
In fact, they say that that's the case here. But why all these other things, right?
But I think this is a strategy to really bias the macromolecular synthesis toward the virus. And that's not that unheard of. I mean, it's becoming more and more of a theme in translation in virus-infected cells that they're doing things to the ribosome. And so this is just a way of,
right.
I think.
Maybe.
I mean, they state that in the article, and I just kind of said, well, duh.
Well, but the thing is,
I guess the concept then would be that the virus is bringing all this machinery, this custom machinery into the cell so that it can stuff the cell's translational machinery for the most part and prevent the cell from translating any of its own proteins to direct everything toward the virus.
What about an enriched cell-free extract for this virus to see if it can make it on its own at that level?
Just add ribosomes?
Yeah.
Ribosomes and an energy generating source.
Right.
Sure.
You can do that with poliovirus. You can make cell-free extracts and program, and they will make particles. Right. But this idea that this is needed to maximize viral efficiency is a good idea, but no one has done it for these giant viruses to take out some of these genes and show that you need them, right? That would be... And they do reference this paper that Kathy is referring to. A Tupanvirus might preferentially target some ribosomes to favor the translation of its own proteins, as previously described for pox viruses. And they reference a paper where the title is Trans-Kingdom Mimicry Underlies Ribosome Customization by a Poxvirus Kinase. Sounds actually interesting.
Mm-hmm. Right, but there's also studies from the herpes viruses, and
but they don't have
others.
But they don't encode their own translation proteins and tRNAs, etc. Right? They do other.
Well,
things.
they're modifying the ribosome.
Yeah, right.
So yeah. To me, it's just different sides of.
Well, maybe it might be. You have to really show it, right? And
yeah. Oh, yeah.
You have to do that. Someone's walking on our ceiling, and Dickson's very worried. He's not going to fall through. Don't worry.
It certainly sounds like that.
It happens very frequently.
It's horrible.
Usually you're sleeping, so you don't hear it.
Bang him with the stick and tell him to shut up. No. Somebody's trying to sleep down here.
Only the ribosome is lacking. The French have a way with words.
Yes.
And they also say the longest tail in the known virosphere. I thought that was a cute one also.
Yes.
Lots of potential titles in this paper. Mm-hmm. So this infects, as we said, two different amoeba, but they say we don't know what the actual real host is of this virus. We have no clues about the hosts of Tupanviruses at their original habitats. I like it, at their... And so, what else? Anything else you want to talk about?
Well, it's interesting that this virus shows up in these two geographically very different environments, even if they're both relatively saline places and they're extreme, but they seem like they'd be extreme in different ways.
Mm-hmm.
And yet we have this same virus turning up in these two places, either because the amoebas are similar enough there or because the virus, I don't know, got from one to the other somehow.
Yeah, it is. It is quite interesting.
And they have this the whole figure at the end showing how the virus seems to have, interesting effects on host cells that are sort of unexpected.
Yeah, you're talking about the tetrahymena effect. Yeah, so it apparently doesn't replicate in tetrahymena, but kills them.
Mm-hmm.
And so they make this statement that this is unprecedented, that a virus would kill the cell without replicating. And so I left a note to Kathy. I said, I thought that happened with adenovirus and fibers. It could kill cells without replicating, right?
Right. And what we know about adenovirus is that when that's happening, you're using probably a really high titer virus stock that probably has even more defective particles in it. And so you're just giving a huge amount of protein. And if you just purify fiber protein and put it on cells, that can be bad for just the cells themselves. But so I had forgotten about this tetrahymena story. I was thinking mostly about is that
that's the one toxicity
section for toxicity where it's phagocytosed in a voracious manner by the tetrahymena. I like that. And so then the figures that they showed in I through K, I don't know. Well, they also talked about UV inactivated particles, and that's where I know that they were using a very high multiplicity of 100. So that's where I got the idea that maybe this is not so physiological. And so I don't know what multiplicity they used for the tetrahymena, which is 8 I through K. So that they're really good about talking about their multiplicities in almost all the figures. But of course, I can't find it for this figure.
So they say they can get toxicity in acanthamoeba at higher MOIs, 50 and 100. So presumably the tetrahymena is lower than 50 or 100. I don't know. I just think that there are other examples where you could add virus and get killing without replication. So it seemed to me their statement that it's unprecedented seems not correct, but I could be wrong.
It wouldn't have the same impact if they said it was precedented.
Well, they don't have to make that statement at all, right? They do say
the point.
To the best of our knowledge. They have a little caveat.
Right.
The best
That's
of
the
our
way
to cover it. Well, I said it was to the best of my knowledge.
Exactly.
And the other thing that I mentioned, I want to just touch on. So during replication, they did some EM. They see vesicles containing ribosomes 2 hours after infection, close to the nuclear membrane. The small vesicles aggregated, accumulating more ribosomes, forming large structures containing ribosomes, which were fully depleted from the amoebal cytoplasm by 6 to 9 hours post-infection. And at that point, they could also see shutdown of ribosomal RNA synthesis. And the nucleus degrades. So it's interesting that they must have all the ribosomes they need by that point, right?
Right, and then they can sort of sequester them from cellular RNAs, perhaps.
Maybe. Mm-hmm. Maybe something's going on in those vesicles. So this is why these big viruses are cool. All sorts of new things that you can investigate. And so there you go. Okay, so one last thought here. So where did these translation protein genes come from? So we think there are two ways that viruses probably evolved, not mutually exclusive. They started small and acquired genes, or they started big and lost what they didn't need. This could be either one, and they say we can't tell if these protein synthesis genes come from a cell or not. We just can't. Look, the tree doesn't tell us that. And I love what they said when they were looking at this. They made phylogenetic trees of the aminoacyl tRNA synthetases. Wait, I'm lost here. Too many figures. Wait. Discovery page 1. It's not page 2.
Sound like Solieri.
Who?
Too many notes.
Too many notes. No, it's not that page. Okay, here we go. Phylogenies based on aminoacyl tRNA synthetases are acknowledged to be complex because they depend on sampling, sex, etc. And they say to reduce such disturbances in the trees during their construction. There was a disturbance in the tree.
Yes.
Too much wind.
Isn't that funny? Disturbances in the trees. I like it. Anyway.
I like it.
Okay. And so this virus could have acquired these genes, or it could have started out with them, leaving the cell and lost them. But we won't know because we can't tell at the moment. Maybe we just have to keep sequencing and find all the relationships. Right. So there you go. Another giant virus. And this one is very pretty.
I just wanted to say they do show a couple of different phylogenetic trees in here. And in figure 3B, they show one for the DNA polymerases. And it's one of those circular kind.
Right.
And then in figure 6F, they show the traditional kind. And even though if you try and blow up figure 6F and read the words, they're hard to read, but I get much more out of the traditional kind. And we'll come back to that.
I see. We're going to come back to it.
Yes, we are.
All right. That's our giant virus of the week. And now it's time to move to bats. Who likes bats?
Bats are cool.
I like bats.
This is a paper in Cell Host and Microbe. It's called Dampened Sting-Dependent Interferon Activation in Bats. And it comes from Wuhan Institute of Virology and Duke National University of Singapore Medical School, which is in Singapore. It's a joint venture with Duke University. And we have Shi Li Shen Go Zhu Kui Lin Fa Wang, the real Batman. He was on TWiV. Shi and Peng Zhu.
There's also a contribution from the University of Chinese Academy of Sciences in Beijing.
Yeah, look at that. Snuck in there. It's snuck. I highlight all the universities beforehand so I can see them, and sometimes I miss them. I just like bats because they look great. I saw a video of one flying the other day. It was so cool. Now, in terms of virology, bats are interesting. Dickson.
Yes.
Bats are the only flying mammals.
Yes.
Correct? 20% of all mammals.
That's true.
They have a positively selected oxidative phosphorylation pathway.
True.
Because of their increased metabolic activity during flying, right?
And
what does
as
this
a
mean?
result...
What does this mean, a positively selected let's deconstruct this for Bob. Or for me.
Yeah, because this is a key part, I think.
It is, because
Brianne,
it
what does it mean?
Go ahead, Brianne.
Oh.
Positively selected oxidative phosphorylation pathway.
So basically, this is a discussion of a type of metabolism that these bats are doing to gain energy. And this is important because it is part of their adaptation to flight. And positively selected means that there have been some genomic studies that have looked at genes, and there seems to be a particular amount of change there. So it looks like this is a unique adaptation in bats.
We did that. We did a genome bat paper a long time ago here where the first bat genomes were sequenced, and they found that there are lots of, I think, duplication of genes involved in OXPHOS. So that's the idea is that you fly, you generate, a lot of oxidative stress.
Takes a lot of energy. Yep.
Plus, it results in a lot of cell destruction.
Yeah.
Yes.
Right.
Yeah,
and
so
release of endogenous DNA.
The metabolism is that you do a lot of damage to the cells, and so you need to also jack up the damage control circuitry.
Yes, although they didn't actually ever show the damage, and I would have loved to see some of that.
So the other part of this coin, or the other side of this coin, not part, is that bats seem to have lots of viruses. This is known, right? There are papers that say, bats seems to have more virus than other species. And are these two things related? The fact that they have to deal with this higher metabolism and they've modified their gene set, does this permit viruses not only to replicate in them, but not to harm them? Right.
Exactly.
And there's reason to think that those two things might be related. The damage that's happening with the oxidative phosphorylation could be DNA damage, and that could put nucleic acid in the cytoplasm of cells, and viruses could put nucleic acid in the cytoplasm of cells. And so these two things could sort of be related, and the bats could have one common mechanism of dealing with that cytoplasmic nucleic acid.
And the presence of DNA in the cytoplasm is unusual. It's usually in the nucleus. And so the cell has ways to sense this and figure out that something bad is going on. And so this leads to a whole set of sensors and innate immune responses. And the big overarching hypothesis for this is that these two types of ways that DNA is getting in the cytoplasm are going to lead to some difference in the innate immune response in the bats.
Yeah.
And so maybe particularly the bats may have tamped down their inflammatory pathways, which would otherwise be constantly activated by this DNA that they've got in their cells in the cytoplasm.
Exactly.
Well, I would say natural selection has tamped it down.
Well, yes, right.
Yes.
When I say bats have done this,
I
what I mean
know.
is through their evolution this has happened.
I just like to be a pain in the neck, that's all.
I know.
It's my role.
Do you think pterodactyls did this also?
Well, I want to know about birds. I mean, birds fly a lot, right?
They do.
Mm-hmm.
Do they have
They
more
do.
viruses than other species? Like bats have more viruses than other mammals. Do birds have more viruses than usual? And if so, has anyone looked at genomes of birds and made? I don't think there's as much interest as here in bats, but maybe that should be looked at, right?
There isn't. There's a little bit more known historically about bird immune systems compared to bat immune systems, but that hasn't really correlated to knowledge about bird innate immunity, at least in my mind.
Yeah.
You'd also have to, I mean, because birds are birds, you'd have to look at flightless birds compared to flying birds.
Yeah.
And those wouldn't even really be that relevant a comparison. What you'd like to look at is dinosaurs.
Yes.
Right.
Yeah, I can do that.
Birds before they flew evolutionarily, and those are all gone.
We don't have a database of dinosaur sequences?
No, unfortunately not
No.
yet. But with bats being mammals, we can look at closely related mammals and say, okay, here are the differences between bat immunity and other mammal immunity. And so when shrews learn to fly, this is the set of adaptations that came with that.
So what are some flightless birds that we could use?
Oh, you got a lot of them. You got emus, ostriches, rheas,
Penguins.
Now,
a ton of them.
Kiwis.
So this is why the malaria in the sparrows infected the penguins in the Bronx Zoo.
That's right.
Because they're both birds.
That's exactly.
Actually, I think a lot of species of rails or subspecies of rails end up being flightless.
All right, so back to bats. DNA sensors. There are lots of DNA sensors. Right. Because as Kathy say, there's not supposed to be DNA in the cytoplasm. Right. So we have sensors to make sure that they're not there. And we have talked about a few of these on TWiV. We have talked about IFI 16. We haven't talked about AIM 2. We've mentioned TLR 9. A few of the others we have not mentioned, DDX 41, LSM 14A. You'll be tested on these later, so remember them. And then
I'm writing all this down.
That's good. I'm excited for the test. My lab works on all these things.
Well, the prize will be a book,
There you go.
Hey, I've already got a book. Okay.
And you got a lot of books. They fill my office, by the way. C-gas. Remember C-gas?
Who could forget?
Cyclic GMP AMP synthase. Jumping C-gas. It's a flash or something.
Yes.
That was the title of our episode.
Like
C-gas is, as they say, the universal and essential DNA sensor. Senses DNA, makes cyclic GMP AMP, which in turn binds and activates Sting, which is not a rock guy.
No.
Although we had an episode called Sting and the Antiviral Police.
Yeah. We did?
That's right. We did.
Yeah.
And then this Sting, once it is bound with cyclic GA, will then interact with a variety of kinases and trigger the production of nuclear genes that are involved in innate responses. And Sting is a cool protein.
No.
I have a picture here.
Sting is a really cool protein.
Sting sits in the ER membrane, right?
It does, but it actually moves
after binding CGAM?
signaling. Yeah. Yes, it does.
So CGAM binds Sting in the ER, and then it moves to the Golgi. And then it interacts with a kinase, with a couple of kinases, right, to lead to upregulation of cytokines in the nucleus. Yeah. So I put a picture. I put a picture in the notes, which is from Principles of Virology. This textbook I found lying on the street this morning.
Is it in the current one?
Yeah, it is.
It must be in volume 2, maybe.
It's in volume 2. Yeah.
Okay, I pulled down volume 1 to look for it. It's a nice figure.
It is a lovely figure. And Brianne, this was made, 4 years ago. Is this still correct?
Yeah, it looks pretty good. I'm still happy with it. The one other thing that's really nice about Sting is that, people can argue about some of the other sensors that are listed in this paper, but one thing that everybody kind of doesn't argue about is that they all signal through Sting. So Sting is sort of a nice central mediator in all of this.
He's the DNA police.
Yeah,
yes.
Exactly.
We're waiting to say that.
I had to.
Okay, now this brings us to the genome of bats. So we're getting more and more bat genomes sequenced. And as we mentioned before, maybe we didn't, but here it is. The most positively selected genes are in the DNA damage, checkpoint pathway, and innate immunity. So flying and viruses, right? Maybe. Mm-hmm. And one of them is a sensor. But what's cool is there's an entire group of genes missing in bats, including these DNA sensors AIM2 and IFI16 that we mentioned. This is called the PIHIN gene family. It's gone. It's gone from the bat genome. They have a contracted type 1 interferon locus.
Right.
And another DNA sensor, toll-like receptor 9, is under more positive selection in bats than in other mammals. So there's something different about DNA sensing and interferon responses. So that's the idea here, right? That this is the crux of the matter.
Right. That is the idea, and it's sort of nice because it, again, ties together DNA damage and viruses.
Yep. So they looked at sting. They looked at all the sequences of bat sting. Bat sting, Dickson.
Right.
Yes.
I heard you.
There's one residue, serine 358. It's in everything but bats. In bats,
it's not a serine.
It's not a serine.
It's a bunch of different things, but it's not a serine.
358. Now, Dickson, where would that be in the three-dimensional structure?
In the pocket of the active site.
Look at that. That was quick. So this site,
fast learner. I just don't read enough.
is critical for interferon activation downstream of sting.
Yes, it's the residue that is phosphorylated by that kinase you mentioned earlier.
Mm-hmm.
Tank binding kinase 1.
Yes,
TBK1. It phosphorylates serine 358, but bats don't have serine at 358. So the other residues, NHF, Y, P, D, and R, those are not phosphorylatable, right?
Right.
So right away you have an idea here that sting might be weakened in some way. So that's where they do their experiments here.
I want to just add that in the non-bats list, they have chicken and zebrafish. So they also have that serine at 358, but bats don't.
Chicken and zebrafish.
Mm-hmm.
Do chickens fly usually?
The wild
fly
ones.
A little
bit, right?
Ones do, yeah.
But the ones in the farms don't.
No.
So I wonder where they got this sequence from. Because there are lots of birds that are flying all the time, right?
Right.
Oh, yeah.
So I just wonder if there's a difference within birds, in terms
of... As I recall, chickens originated from some Indian jungle cocks, as they were called.
All right, so let's compare sting responses between bat cells and other mammalian cells. So they use mice. They have 3 Rhinolophus sinicus cell lines, right?
Mm-hmm.
And they add CGAMP to them.
Yes.
Which is a little molecule, right? Cyclic GAMP.
So this is a small molecule that is made by C-gas. It's basically an AMP bound to a GMP.
Right. And can you buy this?
You can. I have lots of it in my freezer. There are actually a few different forms of it. The GAMP and the AMP can be bound with different chemical sort of bonds.
Yeah.
So
I bet that wasn't such a popular product before Sting. Before
no,
C-gas.
I don't think so.
So funny. Anyway, so they add CGAMP to these cells, and they're measuring interferon beta and IRF7 mRNA, which would be outputs of CGAMP, the signaling pathway. And it's lower in bats compared to mouse.
Mm-hmm.
And the control here is to add double-stranded RNA or sendivirus, which will hit, of course, the RNA sensors. But you get interferon beta and IRF7, and the mice and the bats are the same.
They are. The one thing that would be really nice here is they only show us what happens with these cells when they are stimulated with something.
Mm-hmm.
And I'd love for them to show that bats don't have a higher amount or a different amount of interferon at rest.
Mm.
Right.
If their DNA is being damaged so frequently, then there are ligands that could turn this on. And so I'd like to know that there isn't this interferon production at rest.
Then they also looked at the genes induced by interferons. They did RNA sequencing to look at that. Because as you know, when interferons produce, that then turns on many other genes, and they find these are called ISGs, and the bat ISG upregulation is much less than the mouse, which I guess you would expect with the lower induction of interferon. Okay. Now, next thing they looked at, they did a phylogenetic analysis just to see if this sting is really different from mammalian sting, if it's expressed in different tissues at different levels, and it doesn't seem to be, so that doesn't explain the differences here. Next, they take human cells which do not have C-gas and sting, and they put sting into them from bats and humans together with human C-gas and a plasmid that you can use to assay for activity of interferon beta promoter.
Yes.
We're reconstructing the whole thing in these human cells.
It's a particular human cell that doesn't have C-gas and sting.
I bet you like these cells, Kathy.
Yeah, 293 cells.
Yeah.
Yes. And this is a pretty standard assay that we use and that most labs use to study sting and C-gas and things like that.
And they are immunologically screwed up.
They are very screwed up, yes.
Well, which you'd expect.
Why would you expect that?
Because the whole system's been mucked with, right?
Well, C-gas and sting are missing. Other things are missing, too. They're missing whole swatches of interferon genes, right?
Yes.
As well.
They are great cells for transfection. They can take up DNA very well, and that could be related to their lack of C-gas and sting.
Yeah, could be. But beyond that, if you want to study innate responses, you probably shouldn't be using HEC unless you're going to reconstruct the whole thing in here. But if you're going to just study... And
it's a great model for this because you want cells that lack these components so that you can put in the specific ones that you want to test.
Yep.
Which they do.
So they take human sting and they put an alanine in place of the serine and they add it to this system and that knocks down...
So they create Batman sting.
They create Batman sting. That's right. And then they can also take the bat sting and put a serine back to 358, and that raises the induction of interferon beta.
So swapping this back and forth from a serine or not a serine seems to give you exactly the effect that they're seeing in the bats.
Right. And they also add CGAMP to these cells and show that if you have a serine at 358, you get good induction of interferon. So that all these experiments, and Brianne, tell us if you like them. They say that 358 serine is really what's responsible for the reduced sting-dependent interferon activation, right?
Yeah, I like these experiments. They, especially with the PA cells, they can, get a good amount of interferon coming out of the cells with just that one mutation.
Yeah.
The PA cells are bat cells?
Yes, the PA cells are, I think it's Terapis electo, bat species.
You have bat cells in your lab, Brianne?
I don't. I'd love some, though.
You should get some.
I would like to get some.
Because if you work on, this sensing, you're working on DNA sensing, right?
I am, yes.
Get some bat cells. Linfa will send you some.
Okay, I'll ask.
Tell him I sent you.
Okay.
He was on TWiV. He has to do everything now.
Sounds good. And just Dickson, to answer a question you had a little while ago, I was just looking on NCBI, and the sequence of 4 different bird sting genes is available.
Oh.
So I will be examining that a little later.
All right.
So now the question is, what does this different have? What's the effect of this difference, this S358 or either S or something else? What does it have to do with virus infection? What's the effect? So they use herpes simplex virus and they infect these bat cells. And if you have a human sting, S358, it's more effective, 2.5-fold more effective at blocking viral replication than if you have a 358A. And the bat sting is not as effective, right? Because that doesn't have an S there. So, they say that this is... So it's not good, but it does block a little bit. right?
Mm-hmm.
It modifies it.
Because it would have to, because you can't wipe out the bat, right?
They're virotolerant.
Right. So said another way, because I'm not sure you included the other half of the bat experiment. If you have the human sting with the S at 358, you get less herpes replication. If you have the bat sting, but you mutate it so that it has the S at 358, you also get less herpes replication. Again, showing that it seems to really correlate with the serine at 358.
Right.
The key there, though, is that it's not going crazy.
Exactly. So I have maybe a silly question. Are there a lot of herpes infections in bats?
I seem to remember that there are herpes viruses. From all the metagenomic studies we've looked at, they find lots of herpes virus sequences. But this seems like a human herpes simplex virus.
Right.
And I'm surprised it can infect bat cells, but maybe it has a broad tropism.
I'm finding
maybe they picked it.
Well, there's one paper that apparently hit the news in 2016 that new herpes virus in bats. So
the thing is, Brianne, you might want to have a bona fide bat virus here, right?
You might. People talk about so many different zoonotic viruses that might have bats as a reservoir.
Right.
It might be interesting to see one of those viruses here.
Yeah. So the key is that the idea is that this sting change will reduce the response to a virus infection, but not eliminate it, because you still have to control the virus, right?
Right.
Mm-hmm.
And you have to still deal with flying and all the oxidative damage that's causing as well, right? So it's kind of a balance between the two, I suppose.
Exactly.
I guess the last experiment, they looked at phosphorylation of these downstream proteins downstream of sting, right?
Mm-hmm.
So they again used their 293 cells. They put human or bat plasmids producing human or bat sting. They find that phosphorylation of IRF3 but not TBK1 is higher when you have S358. And that, I guess previously others have shown that this residue 358 is important for IRF3 but not TBK1 binding.
Right.
So that's consistent with that.
And that's a nice way to show that this is sort of, dampening the response or shifting the response, but not getting rid of it entirely.
Yeah. So this phosphorylation of IRF3, that is the protein that binds what's it, phosphorylated by TBK1? Is that right?
It can be. IRF3 is really a big transcription factor.
Yeah. That's
leading to the transcription of interferon.
But it needs to be phosphorylated to get in the nucleus, right?
Yes.
So does it? I suppose TBK binds sting with CGAMP in it. That activates TBK, which will then phosphorylate IRF3, and then it goes in the nucleus, and you get all this. So what they're showing here is that phosphorylation of IRF3 is affected by whether you have a serine or something else on sting, but not TBK1.
Right.
Which is interesting, because TBK1 is supposedly interacting with CGAS sting, with the CGAMP sting.
So another unrelated question, of course, from left field. Speaking of bats, do bats transmit viruses vertically as well as horizontally?
Vertically, meaning
a baby bat is born. Yeah. Where do they acquire these viruses?
I assume there must be at least some that are transmitted vertically in bats.
I mean, I think about rabies as a bat-associated virus that's really horrible for people, but not for bats. So that we can understand how that occurs now. But that's sequestered away in the nervous system, isn't it? Isn't that a neurotropic virus in bats? I don't know, but I'm making an assumption here.
So I found an article that says that Nipah is vertically transmitted in bats.
Okay. Because, there are regions in the world where...
cats.
Cats, not bats?
I searched for bats and I got cats.
Oh,
I also got the
rats.
top hit. I searched for vertical transmission in bats and I got a trematode article. Now you go figure that.
Not me. I know what a trematode is, though.
So here, sporadic vertical transmission in bat pups has been observed. Yes. Okay. So the answer is yes. I didn't know the answer to that.
Yeah.
So their summary is very nice. These results demonstrate that while bats sting, maintains its antiviral defense similar to human sting. The dampening likely contributes in part to the long-term coexistence of bats and viruses. I think that's a lot to say from...
A lot to say.
I mean, I think
Sure.
this shows that it's dampened and that it can still pretty much control the herpes simplex virus infection, but
Yeah.
I don't know what experiment you would do to actually address this, because it's not something you can do in a couple of weeks.
No,
Right.
not at all.
Long-term coexistence, right?
Yeah.
Mhm.
But you could, I don't know if you could do any knockouts in bats at all, right? That would be interesting to do, or knock-ins. If you put in this 358 with a serine into bats, what would happen?
Do they suddenly get sick from a bunch of viruses, or do they suddenly stop sustaining the viruses,
Or
what?
when they fly, do they drop dead because they have too much DNA damage?
Right. Do they suddenly have all kinds of metabolic problems?
When I talked to Linfa, he said there's they have a colony of bats that they're studying. He says, but the problem is we don't let them fly around. So it's really artificial. They're sitting in these little cages, and we can't ever assess what anything we do to them has on their consequence of flying, right? Because you can't. If you fly, they'll go away, and your experiment's out the window, so to speak.
Right. And you couldn't have a big enough, you'd have to have a huge outdoor enclosure where they could fly around.
Yeah.
Huh.
So
I guess you could try putting them in a wind tunnel, but that seems problematic.
This looks really good, but I think it's going to be hard to prove.
Yeah. It's a tough system to work on.
I don't know, maybe you could knock in a S358A into mice and see what happens there if they have an over-exuberant... If I guess virus infections might overwhelm them or something. I don't know.
Well, you could knock it into pigs and see if they can then fly.
I knew you were leading towards that. I didn't know how you were going to actually work it in, but I'm still stuck on a concept here, though, that baffles me a little bit, because most of the bats that we're used to seeing with people, like, for instance, the Australian bats, they're all fruit bats. They're, we're not dealing with carnivorous bats here. We're dealing with fruit-eating, peace-loving mammals that have no interest whatsoever in transmitting viral diseases to humans. But
nothing has interest in transferring viral diseases to humans.
No, I agree with you, Alan. But I'm just saying that the ecology of this.
Right.
So where do these bats pick these viruses up then?
In their roosting colonies.
They exchange everything.
Each other.
They do.
They're pretty social.
They're very social.
So they're
extremely social.
They give it to each other. Yeah.
But you had to start somewhere, though.
Yeah, how did the first bat get it?
Wow.
Well, you could ask that about
anything. Yeah. That goes way back. They've been around as long time.
They have.
For doctors of philosophy to figure out.
Yes.
Right.
Well, we could figure it out, right?
Yeah.
Come on, we've got PhDs.
That's why I said it.
Yeah.
So Brianne, you work on sting and sea gas?
I do. I work on a few of the sensors that are mentioned in this paper. In fact, I started the lab looking at IFI-16.
This cytosolic DNA sensor, right?
The cytosolic DNA sensor, yes.
And what are you asking about IFI-16?
So we are looking at interactions between IFI-16 and CGAS to see how they and some other members of the pathway interact. And we're also looking at how they may have evolved in non-human primates that don't get pathology from an SIV infection.
So you think it might be dampened?
Exactly, which is why I was so excited about this paper.
I see. So is IFI-16 one that's antagonized by a herpes viral protein?
It is.
Which one is it? Which protein?
I think there are actually a few. I'm not sure off the top of my head.
I think we did one of those papers on TWiV. And there's this other thing that it might shuttle into the nucleus, right?
Yes, exactly.
And sense there as well, because that's where these viruses are going, yeah.
There's some pretty good evidence that IFI-16 spends a fair amount of time in the nucleus. It has 4 nuclear localization signals and localizes to the nucleus quite a bit.
I like it. Spends a fair amount of time. It's like Dickson spends a fair amount of time in rivers.
I do. I do. But there are receptors for that,
So that's interesting twist, that looking at different animals who have different responses to virus infection. So it's not just bats that are going to get all the attention.
Right, exactly.
Cool. Neat. All right. Anything else before we move on? That was a cool bat paper. Yes.
Yeah, I wanted to point out that it was shorter than our snippet in terms of
Yes.
space in the journal. And it only had 2 figures, and then it had 2 supplemental figures and
Yes.
2 supplemental tables. It could have been all in the paper.
Yeah.
Right.
So
yeah, this is in Cell Host and Microbe. This is a brief report, and by the way, this uncharacteristically for Elsevier is also open access.
That is uncharacteristic. That's exactly
So maybe
right.
the brief reports have a figures limit or something. But
Yes, I'm sure they have some strict limitations on that.
But this labeling of brief reports, it's only because of the number of figures. It has nothing to do with the
important
impact,
Yeah,
right?
that's
right.
So that's why I didn't say at the onset that this is a brief report, because
A brief but interesting report.
I think it.
A brief report, but we were not going to be brief about it.
No.
Oh no, not at all.
No,
This
it's
is really
not at all.
cool. And in reading this, there are some other cool papers that have been published on bats in the meanwhile as well. Very
Yes, I
interesting
was
stuff.
circling quite a few things that I wanted to look into.
Very cool. All right, let's do some email. Bowden writes, now this email came in January to my Columbia account, so I missed it. And it refers to a paper that we did a couple of TWiVs ago. He writes, Dear TWiVers, immuners, twimmers, twippers, and tweevors. So I guess that's why he sent it to my Columbia account, because he didn't know which Microbe TV. I listen to all of you every chance I get. I listen to some of your podcasts twice. You have broadened my biological horizons immeasurably. You, like no one, make the world aware of all the possibilities brought to us by biology. The enclosed article with commentary, not in a CNS journal. It's not central nervous system.
Cell Nature and Science.
But I like that. Caught my
yeah.
attention. This article and its commentary may meet your standard for pick of the week. It may even be worthy as an article to podcast. The potentialities of DNA and RNA continue to amaze me. And he sent the article on the endogenous viral elements in the mosquito genome that we did a couple of weeks ago. Thank you for your attention. I am writing from the Health Sciences Library at the University of North Carolina School of Medicine, where it is currently a surprisingly brisk 43F. When it is cold, I use Fahrenheit to feel warmer, and when it is hot, I use Celsius to feel colder.
That's good.
It's very nice.
That's a good plan. I think I'm going to go with that.
I like
Yes.
it. I like it.
Let me take Anthony's, because this is short. He sends a link to an article about harbor seals in Liberty State Park. And the link is to a Facebook page of Jersey City. Jersey Shitty. I know I said Jersey Shitty.
Sigmund Freud is listening. Careful.
And it's a movie of harbor seals. Yeah, there it is. There's a harbor seal. So Anthony wants to know if anyone is watching for seals that might be sick with influenza. Yeah, I think people do look for that. But I don't know if they sampled them. I think they wait for them to get sick and then they sample them, because seals can die from influenza. If anyone is listening who knows all about seals and influenza, tell us what they do. If there's a surveillance scheme in place, or if it's just, reacting to.
That would be good. We have a bunch of them up in Cape Cod.
You do?
As well, yeah.
Alan, could you take the next one?
Sure. Matt writes, Howdy, TWiVumvirates. LTLFTW here. I believe that's long-time listener, first-time writer.
Wow, very good.
Not sure if someone's already used that acronym or initialism to be pedantic. See if you can figure it out. I don't have it. We did figure it out. Thank you. I don't have any specific comments, as I sadly don't get to listen to your show regularly anymore, but I wanted to express my gratitude for what you guys do. I believe science communication to the general public is perhaps tantamount to the science itself, especially in today's socio-political environment, and it is inspiring that you and the other hosts on the Microbe TV world have taken on this role in your busy lives, going strong with still new shows. I'm a first-year graduate student at Johns Hopkins School of Medicine doing my PhD in human genetics, but I've been listening to your show since Dr. Chris Sullivan over at UT Austin put me on it. Hi, Chris, if you're listening. I was actually at your live show, TWiV 255 at UT, too, in October 2013, as I was a wee undergrad taking Dr. Sullivan's introductory course in virology. My apologies, I'm just now reaching out 4.5 years later. After my PhD, I wished to stay in academia, but I've toyed with the prospect of having a career or at least some involvement in science communication since I've been listening to your shows. I'd imagine there are many overlaps with the traditional university lecture, and I'm sure you can comment on this. In any case, you might just get a request from me for insight and wisdom on your experiences in this line of work many years down the road from now. If I may submit a listener pick, my favorite TWiV episode 443 on a leaf no one can hear you scream to those who haven't yet listened. The findings discussed here are a hauntingly beautiful example of the driving force viruses can play in parasitism, and it's just phenomenal. Finally, if you're still accepting CD4 Hunter scores for the Hotez book giveaway, I've attached my screenshot. You got a score of 4680. I hardly think it will be at a highest, but I was surprised to learn that one could get such a thing as phone screen finger burn from constantly shaking off those darn antibodies. I got to a point where I had to alternate between my pointer and middle fingers, and at times even my thumb. Thanks again to each of you all and your guest hosts for everything you do.
Thank you, Matt.
Thank you.
All right, so let's do some picks of the week. Alan Dove, what do you have?
I have another education pick, continuing on an arc, I guess, from last week. This is a program called Letters to a Pre-Scientist. And I'm really picking this not so much to get more scientists involved because they have an overabundance of scientists who volunteered for it. Although if you want to, you certainly can. But I'm hoping that, I know we have some teachers who listen to this podcast. I'm hoping some of them will get involved with this and sign up more students for it. The concept is that it sets up a pen pal relationship between students, generally in secondary schools, and scientists, working scientists. And so the idea is that the scientist writes letters to somebody who wants to be a scientist about what they do. And the students write back and ask questions, and you correspond.
It's cool.
That's great. I really like that.
I think it's a lovely idea. And you can sign up right on there. You join their email list or volunteer with them. Or I'm sure if you wanted to donate money, you can do that. And as I say, they do say that they have quite a few scientists, more than they actually need. But if you are a scientist and you want to sign up, then please do so. And if you're a teacher, please really look into this, because I think it sounds like a really, really great program.
There was some kind of program like this when I was an assistant professor, and they would send kits of stuff to the schools. I don't know if that's what's involved here too. And then they would match you up with a scientist who would also get a kit and know what the exercise was, and they could write letters and back and forth. It was
they have lots of pointers and advice for both sides of the conversation.
That's great. Love it. Brianne, what do you have?
I have an article from CIDRAP, the Center for Infectious Disease Research and Policy. And I found this really interesting because when I'm talking to people about vaccines, I often hear people say, well, I just think it's too many antigens. It's too many vaccines all at once. That seems to be a common complaint. And so this is actually reporting on a study that just came out in JAMA, where different groups of kids with different different diseases were examined to see how many vaccine antigens they had been exposed to see if, in fact, number of vaccine antigens was related to having infections or having different pathologies. And in fact, they saw that there were no differences in antigen exposure between those who were diagnosed with the illnesses and those who were not. It was actually 240 versus 242. So it looks like this argument that too many antigens or antigen overload is not a problem with vaccines.
I think it's great that they did this.
Yes.
However, it's a dumb argument to begin with, right?
Oh, I agree. But I often find myself having to argue for vaccines on this.
But amongst us, this is ridiculous. I think it's too much, really. Now, what do you base your thinking on? Have you really looked into how much antigen you get versus a real infection? I mean,
or how much antigen you get just in everyday living.
And how much antigen a baby crawling on the floor stuffs in their mouth.
Exactly.
I mean, the concept that kids are being exposed by too many antigens through these very precisely targeted vaccines is absurd on its face. But as you're pointing out, Brianne, it's good to do a study like this when this argument is being floated so consistently by the anti-vaccine folks, and this is one of the big tropes in the anti-vaccine movement is, oh, it's too much, it's too soon, it's too many vaccines at once, yada, yada, yada. So to actually really, really blow that away, it's nice to have a data set you can point to and say, look, definitively, it is not a problem.
Right, exactly. I just like having data to be able to help to address this argument.
They will criticize this study.
Oh, of course. But only the critics of this will be the hardcore folks who you're never going to reach.
Mm.
This is for the sake of the folks Brianne is talking about, and I'm sure, I've had similar experiences, and anybody who raises kids these days has similar experiences where the conversation turns to vaccines, and inevitably this comes up. And it's generally not some hardcore anti-vaccine nut you're talking to. It's just somebody who was not trained as a scientist, and they're trying to figure out the best thing to do for their kid. And this is a great thing to point to say, yeah, the schedule is fine. It's not too many antigens. You can ignore that argument.
Exactly. It's probably the most common thing that I hear about when I tell someone I have a PhD in immunology.
Yeah.
Dickson, what do you have?
Well, I have what I thought was a simplified flow chart of virus families and the ones that are involved in vaccine development, which to follow up on.
Vincent is now picking on.
Yeah, but that's okay. And rightly so, because they did misspell a few words. It wasn't carefully checked, I think, by virology experts. But it was published in the American Scientist, and it was an attempt to show the broad spectrum of viral infections and the vaccines that have been created from them. And it's a good summary of someone that's not a virologist, but is interested in knowing what state of the field of virus vaccine development there is. This is a good thing to go to, I think. So
here's another one, the strains of herpes virus.
Yeah, I know.
The hell is that?
Listen, you want to take it off? Fine, I'll go back to my picture.
I'm just picking on you. It's fine. I appreciate that you're engaged. It's fine.
I am engaged. I'm actually married.
It is a very pretty graphic, and it is mostly structured about right.
Thank you.
Although you can get the same information at...
I actually didn't make this.
What is it? Viral Zone.
I don't,
which is a great site.
Maybe American scientists can modify it and upload something better.
Yeah, they should fix it.
Perhaps.
Go ask a virologist. Ask Dickson.
No, don't do that.
Kathy, what do you have?
I picked something that I asked a couple colleagues for this morning. If there was a way to see how you go from a standard phylogenetic tree to those circular things that I dislike. And so I put in a link to a site that I think is really helpful. And because it shows a fairly simple standard tree, and it even has viruses on it for its example. And then it shows you the circular type and then the other type. And they have the tree that's I call circular is in polar format. And it tells you that the horizontal dimension is the distance from the center point, and the vertical dimension of a standard tree is now the angle of the circle. And they say, these trees are generally used to make a big visual impact in papers, but generally have reduced readability. It is difficult to compare how far nodes are from the center.
Right.
Generally best avoided. And then they talk about a radial tree, and these are the ones that kind of just, once you see this radial tree, you'll understand it's just lines, and they're not in a tree shape. And it says this format tends to clump closely related sequences together, making their precise relationships difficult to see. Generally best avoided too. I will not mention these formats again. And then the webpage goes on to explain it. But it is kind of nice to see how a regular tree gets converted into a circular tree. But of course, their regular tree is simple, and so their circular tree is pretty simple. And we're never faced with those in the journals. But as I said, in the first paper that we had today, they had a circular tree and then they had a linear tree. And even though the linear tree was almost illegible if you tried to blow it up, I liked it much better. But if you want to see how these convert, this is a helpful site.
Thank you so much for posting this, Kathy. I'm pretty sure this is going straight into one of my lectures.
So this is by Andrew Rambo. And when we wrote the last edition of Principles of Virology. I got his permission to put this in the book. So it's actually in volume 2.
Oh, good.
Principles of Virology. It's in a condensed way, which is maybe a little more digestible, but it's very good. Yeah. I have 2 picks, 2 articles on 2 things that we've been talking about lately. The first is in Science-Based Medicine, which is really an awesome site. It's called True Believers, Entrepreneurs, and Scammers in Alternative Medicine. Great article on these 3 classes of quackery. Alternative medicine, homeopathic, blah, blah, blah. There are 3 categories of quackery: true believers, entrepreneurs, and scammers. There is considerable overlap, and it's possible for one source to be all 3 at the same time, but usually one category predominates. And it goes through examples of each, like one young lady, a true believer killed by alternative medicine, and a person with an online presence, a quack entrepreneur, people who post stuff that's just wrong. Heal your cancer naturally, stuff like that, right? And these people make videos, and they have a huge presence because people, I don't know, they get sucked into it. They have one person, Mike Adams, in a category all his own. Anyway, if you want to be disturbed about what the internet can do, video seems to rule these days, and no self-respecting quack would be without a YouTube channel. Complete with videos, they could also post on their Facebook page. The more complete quacks even have documentaries on which they build their brand. And Vax, the movie, is an example of that. Not only is there the movie, but there's an anti-vaccine Facebook page, a YouTube channel, to which the film's producers Del Bigtree and Polly Tommy, anti-vaccine activists both, regularly post videos which range from them ranting about vaccines to them showing interviews with parents who mistakenly believe that their children were vaccine injured. Occasionally, even Andrew Wakefield himself deigns to show up. It's just disgusting, absolutely disgusting. But that's where we are. And the other article is the other side. It's a very nice commentary in PNAS by Walter Orenstein and Rafi Ahmed. It's called, Simply Put, Vaccination Saves Lives. And they have lots of information about, how vaccines work and how they have worked over the years. And I know Alan would say, this doesn't work. You can't give them more information because they'll just say it's wrong.
It doesn't work on the hardcore, the true believers from the previous post.
Right.
This can work on the folks who,
like Brianne, like in Brianne's case, right?
Yeah, and like I've talked to who just don't know, and they hear different things and they say, "Oh, well, is this a controversial issue?" And you can point to something like this and say, no, this is not a controversial issue. This is an issue where you have very good, solid data on one side and a scam on the other.
Yeah, and this is open access, so you can send it to people. I mean, let's put it straight. My other pick is about quacks who want to make money by doing this.
Right.
I don't think they have any idealistic viewpoint. They just want to make money. And they found that this does it. They can sell crap to people, and they're doing it, and they don't care that people are going to die as a consequence. They really don't. And so this, if you want some facts to give people, really good numbers. We had already 2 picks. One more from Vinayaka. Hi, Vinny, because he calls himself Vinny. Greetings from Einstein. Considering all the science education and advice to young scientists that you are podcasting these days, I thought this was an apt pick for your listeners. It is a website called Code of Ethics for Young Scientists from the World Economic Forum. So this is a code of ethics for researchers. And they explain what it is, the principles that are involved, like minimize harm, pursue the truth, support diversity, be a mentor, etc.
Engage with the public.
Engage with the public, which we do, right?
Yeah. This is good.
It's really cool. Very nice. By the way, I listened to the Immune podcast, and I'm also hooked. Both Cindy and Steph are fabulous co-hosts, great choices. So Vinny is a professor at Albert Einstein College of Medicine in the Bronx, not too far from here. And then we also had picks from 2 others who I can't remember, but they'll be in the list of picks, weekly picks, and that will do it for TWiV 484, which Dickson says is a nice number.
Like it.
You can find us at microbe.tv/TWiV. And if you listen on a podcast app on your phone or tablet, just subscribe, please. Subscribe to us, and you'll get every episode. And even if you don't want to listen to every one, they'll be there in case you have some time. Just do that. That would be great. And if you like us a lot, then give us a contribution, microbe.tv/contribute. Help us run the show. Brianne Barker is at Drew University, and she's at BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
And you wanted to do this paper.
I did
last time, and you got it.
I was looking forward to this paper a lot.
We saved it for you.
Well, thank you.
You're welcome. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And I noticed no snow coming down.
Very good.
It was more than 46 minutes later.
What do they know?
Here the sun's come out, right?
Oh, no, it's nice out there right now. At least it looks nice. I think it's a little coolish, but it looks nice.
Dickson Despommier is at thelivingriver.org and parasiteswithoutborders.com. Thank you, Dickson.
Oh, you're welcome.
I bet next time you're going to have a pick, aren't you?
I'll have a better pick. How's that?
Related to what you do, right?
Well, it could happen. Could happen.
Thank you, Dickson.
You're welcome.
Alan Dove is at turbidplaque.com, and he's Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Isn't it nice that you don't have to have underscore Alan Dove?
Yes.
Some people whose names are taken, they put underscore.
Yep.
Underscore Dickson. You could be underscore Dickson Despommier.
Yeah, you used to always fall
because your name is taken, right?
I used to fall short.
You could be real Dickson Despommier.
You know,
fake Dickson Despommier on Twitter, you know.
Good for them.
Who is masquerading as you.
Good for them.
You don't care.
I don't care.
What do you care about?
Oh, that's another subject.
Vertical farming.
It's another subject.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music, ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.